Hi everyone, welcome to my channel. Today's topic is microscopes. Microscopy is the science of observing objects too small to be seen with the naked eye. From the earliest light microscopes of the 17th century to today's advanced electron microscopes, this technology has evolved dramatically. In pharma, this evolution has transformed drug development, quality control, and even disease diagnostics. Principles of microscopy. The functioning of any microscope is based on three core principles: magnification, resolution, and contrast. Magnification. Magnification refers to how much larger the microscope makes the object appear. It is determined by the objective and eyepiece lenses. Resolution. Resolution is the ability to distinguish two closely spaced points as separate entities. It depends on the wavelength of the illuminating source, light or electrons, and is a critical factor in determining image clarity. Contrast. Contrast enhances the differences between structures in the specimen, often through staining or the use of different optical techniques. Types of microscopy. Light microscopy. It is the most common type used in pharmaceutical labs. Light microscopes use visible light to magnify samples up to 1,000 times their actual size. They're essential for tasks like examining cell cultures and tissues, checking for contamination, and monitoring chemical reactions. This allows researchers to ensure that their drugs are produced under the safest and most effective conditions. Phase contrast microscope. This type allows scientists to examine live, unstained cells, perfect for observing how cells behave in real time. This is crucial when studying the effects of drugs on living cells, offering a clearer picture of how potential treatments will work in the body. Fluorescence microscopy. It is another game-changer in pharma research. By tagging molecules with fluorescent dyes, scientists can track specific components of a cell, such as proteins or DNA. When exposed to ultraviolet light, these dyes glow, revealing important details about how drugs interact with cellular structures, helping in drug development and targeting therapies more effectively. Calibration of microscope. Calibrating a microscope ensures that the measurements you make using the microscope, such as the size of cells or particles, are accurate. Here's a step-by-step guide on how to calibrate a microscope properly. What you'll need: microscope, the one you're calibrating. Stage micrometer, a slide with a precise scale, usually 1 mm long with 100 divisions, each division being 10 micrometer. Eyepiece reticle, graticule, a glass disc with a scale, placed inside the microscope eyepiece. Calculator, to work out the calibration factor. Steps to calibrate a microscope: install the eyepiece reticle, graticule, if not already present. Place the eyepiece reticle inside the eyepiece of the microscope. This graticule will serve as your reference scale when viewing the sample. Place the stage micrometer on the stage. Place the stage micrometer on the microscope stage. This slide has a precisely known scale etched on it, typically in micrometers. Focus the microscope. Focus on the stage micrometer using the objective lens you wish to calibrate. Make sure the scale on the stage micrometer is sharp and clearly visible. Align the reticle with the stage micrometer. Align the scale of the eyepiece reticle with that of the stage micrometer. You may need to rotate the eyepiece to get the lines to line up perfectly. Count the divisions. Count how many divisions of the eyepiece reticle fit into a known length of the stage micrometer. For example, you might count how many reticle divisions span 100 micrometers on the stage micrometer. Calculate the calibration factor. To find the calibration factor, use this formula. Record the calibration factor. Write down the calibration factor for each objective lens, e.g., 4x, 10x, 40x, etc. You will need to repeat this process for each lens as they have different magnification levels and, thus, different calibration factors. Measure objects using the reticle. Once the microscope is calibrated, you can use the reticle to measure objects in your samples. Simply count the reticle divisions that span the object, then multiply by the calibration factor to get the actual size of the object. Thanks for watching. If you found this video informative, don't forget to like, share, and subscribe for more videos.